My name is Zenah, and I am presenting a paper review on the article titled Repeated Cortico-Striatal Stimulation Generates Persistent OCD-Like Behavior by Susanne E. Ahmari et al. This paper analyzes how the activation of a specific cortico-striatal circuit gives rise to OCD-like behaviors in mice. OCD, which is obsessive-compulsive disorder, is a psychiatric disorder in which unreasonable thoughts and fears, which are labeled as obsessions, lead to compulsive behaviors. Previous research has implemented that there is a relationship between cortico-striatal stimulation and the activation of OCD-like behaviors. However, the exact relationship between the circuit activation and behaviors remains elusive. This paper aims to answer the question of, is the relationship due to a direct stimulation and hyperactivity of the orbitofrontal cortex ventromedial striatal pathway, or is it a result of homeostatic adaptive mechanisms that compensate for the abnormalities? This paper utilizes an optogenetic-focused design combined with Krelok's technology to induce long-lasting plasticity that alters connectivity within the circuit. A synthetic Cre-dependent virus that codes for channel rhodopsin fused to enhanced yellow fluorescent protein is injected into the orbitofrontal cortex of EMX Cre mice. After the fiber optic cable is surgically implanted into the ventromedial striatum, various in vivo recordings are performed to ensure that the virus is distributed as intended and is functioning properly. Once confirmed, animals are sacrificed for in vitro analysis of brain tissue. The second phase of experimentation focuses on a behavior-oriented design to understand whether the activation of the cortico-striatal circuit gives rise to a hyperactive grooming behavior, which is an OCD-like behavior seen in mice. The animals are subjected to a series of real-time stimulations while in the open-field apparatus. Various cohort studies with minor changes among each study are implemented. The active virus previously mentioned is injected into the orbitofrontal cortex of the experimental group of EMX Cre mice. The control group, on the other hand, receives a control virus that does not code for the active channel rhodopsin channels. The fiber optic cable is surgically inserted into the ventromedial striatum. In order to habituate, mice are placed into the field without cable stimulation for 15 minutes a day for 7 consecutive days. This is done in order to acclimate them to the field, the fiber optic cable, and experimental handling. On days 8 through 12, mice are placed into the open field for 15 minutes a day. During the first 5 minutes, the pre-stimulation period, the cable is switched off. During the second 5 minutes, the stimulation period, the cable is switched on and CS neurons are activated. During the last 5 minutes, the post-stimulation period, the cable is switched off. Grooming behavior is recorded for the entire 15 minutes. Results show that the control group did not show any significant changes in behavior. The experimental group, on the other hand, displayed drastically heightened levels of grooming during the pre-stimulation period on days 3 and 5. No changes in behavior were seen during the stimulation and post-stimulation sessions. The second experiment uses a timeline similar to the one previously mentioned, where 7 days of habituation are followed by 6 days of stimulation using the same stimulation cycle. However, in this case, grooming data is recorded for a 5-minute duration 1 hour after the 15-minute light cycle ends. Results show that the experimental group indicated a steady and significant increase in grooming behavior over the 6 days of stimulation. This indicates that selectively activating the circuit alters plasticity to induce grooming behavior. The control group, on the other hand, showed no significant changes in grooming behavior. When the behavior of the second experimental cohort is measured again 2 weeks later, results show that the experimental group, positive for active channel rhodopsin channels, continue to demonstrate elevated grooming levels despite the absence of stimulation. The first cohort of the third experiment undergoes a week of habituation followed by a week of stimulation. For 2 weeks after baseline stimulation, fluoxetine is administered to both control and experimental groups, and open-field stimulation is continued. During the final washout week, open-field stimulation is continued and the fluoxetine treatment is stopped. Results show that during the baseline week, the experimental group with the active virus shows elevated levels in grooming behavior. However, after 2 weeks of pairing the circuit stimulation with fluoxetine treatments, there is no difference in grooming behavior between the two groups. During the final washout week, the experimental group with the active virus displays an increase in grooming behavior similar to what was seen in the baseline week. Data indicates that the fluoxetine imposes a mediative effect on the hyper-grooming behaviors of the experimental group with the active virus. The second cohort of the third experiment consists of 2 experimental groups that receive the active virus and undergo a similar experimental timeline used in the first cohort. However, no washout week is implemented.
During the 2 weeks of circuit stimulation and drug administration pairing, fluoxetine treatment is administered to one group and a vehicle is administered to the other. Behavioral results show that during the baseline week, both groups displayed elevated levels of grooming behavior. However, after 2 weeks of drug administration and stimulation, the experimental group receiving the fluoxetine treatment displayed lower levels of grooming when compared to their vehicle counterparts.
To summarize research findings, the first experiment shows that consecutive days of circuit stimulation does indeed influence grooming behavior. The second experiment indicates that selectively activating the circuit to alter plasticity induces a grooming behavior that lasts for days after stimulation has ended. And finally, the third experiment shows that chronic fluoxetine treatment can reverse the increased grooming behavior seen when the circuit is stimulated. In conclusion, the relationship between circuit stimulation and OCD-like behavior manifestation is a result of repeated hyperactivation that causes a change in plasticity and is not due to acute stimulation. The author's conclusion also provides substantial evidence in regards to being able to mediate behaviors through the administration of fluoxetine. Previous models have proposed that specific brain circuits are responsible for specific types of obsessions and compulsions. Because OCD is a heterogeneous disorder, the study may only be of use for particular subtypes. One question that remains unresolved is if fluoxetine treatment can be used to alleviate symptoms in all subtypes. One major impact on understanding human disease and treatment is the clinical impact. If the specific circuit in question can be understood and entirely detailed, further research may allow experimenters to translate treatment methods to become applicable to humans. Fluoxetine administration then may be potentially administered with the intent of easing OCD symptomology in humans.